Good day. We are the Group 4, and today we will be discussing all about the modern drug administration technology of the CRISPR based CCR5 gene editing for HIV. Let's get started. So, HIV therapy today depends on lifelong antiretroviral drugs. While effective, these treatments come with challenges such as strict adherence, risk of drug resistance, toxic side effects, and high costs. These issues show the need for new strategies that are precise, long lasting, and potentially curative. CRISPR gene editing technology provides a promising solution. It allows us to modify specific genes, specifically CCR5, which HIV uses to infect immune cells. This targeted approach can improve treatment adherence, lower side effects, and directly address the root cause of infection rather than just managing symptoms. It also represents a step towards therapy that could one day cure HIV. In this session will help us understand how CRISPR works as a drug delivery tool, explore the mechanism of CCR5 disruption, and examine both the advantages and limitations of this technology. Finally, we will discuss how these innovations relate to clinical and nursing practice, including patient care, education, and ethical considerations. Understanding these advancements prepares healthcare professionals to support patients in the evolving landscape of HIV treatment.
So we'll be talking about the overview of modern drug delivery technologies. So we have here improves drug effectiveness and safety, enhances targeting and control of therapy, reduces dosing frequency and side effects, basis of advanced therapies such as CRISPR. So modern drug delivery technologies improves how medications act in the body, making treatment safer, more effective, and more precise. So they also enhance targeting and control therapy, which helps reduce unnecessary drug exposure to healthy tissues. These systems reduce dosing frequency and side effects by maintaining stable drug levels in the body. Overall, they serve as a foundation for advanced therapies like CRISPR-based gene editing. Now on to our next slide, we'll be talking about the classification of drug delivery technologies. So, we have here controlled release systems, targeted delivery platforms, smart or responsive systems, biotech based delivery systems. Modern drug delivery systems are grouped based on how they control drug release, targeting responsiveness, and biological interaction. So basically, they are classified into four major groups, such as what I have stated there in the bullet form. Yes. Controlled release systems focus on regulating the time and rate of drug release, targeting delivery platforms deliver drugs directly to specific cells or tissues. Smart or responsive system releases drugs in response to body signals. Lastly, biotech-based delivery systems use biological tools such as genes, RNA, and viral vectors. Next, onto our next slide, we have the control, release, and targeted systems. So, as we have here, the control release basically is focusing on the sustained drug delivery, maintains therapeutic drug levels, and its targeted delivery are the specific cells or tissues in our body, and lastly, minimizes systemic exposure. So, these systems improve treatment effectiveness by controlling drug release and avoiding unnecessary distribution in our body. Okay? So controlled release systems allow drugs to be released slowly and continuously over time, which helps maintain therapeutic drug levels. This reduces the need for frequent dosing and improves patient compliance. Targeted delivery systems focus on delivering drugs to specific cells or tissues. As a result, they minimize systemic drug exposure and decrease adverse effects. Last, next slide would be smart and biotech based systems. Smart systems respond to body signals triggered by pH enzymes and inflammation. Biotech systems act at a molecular level, includes mRNA, viral vectors, gene editing. So, smart and biotech based systems provide precision therapy by responding to biological signals or altering molecular pathways. So these systems release drugs only when specific conditions are present. Biotech-based delivery systems work at the molecular or genetic level. Examples include mRNA technology, viral vectors, and gene editing systems like CRISPR. Next would be examples of modern delivery technologies. Nanotechnology or nanoparticle drug carriers, transdermal systems, skin patches, implantable devices, long-term delivery, gene editing carriers, viral vectors, LNPs. These technologies improve how drugs are absorbed, maintained, and delivered for long-time treatment. Nanotechnology uses nanoparticles to carry drugs more efficiently and improve targeting. Transdermal systems deliver drugs through skin patches. So these are common, right? We all know drugs that, that would use skin patches, avoiding gastrointestinal side effects. Implantable devices provide long-term drug release for chronic conditions. Gene editing carriers such as viral vectors and lipid nanoparticles are used to deliver CRISPR into cells. So we'll be tackling about the relevance to CRISPR-based therapy. So this enables targeted cellular delivery, allows gene-level molecular action, improves precision and safety, supports CCR5 gene editing for HIV. These drugs delivery technologies make CRISPR therapy possible, safe, and effective. These modern drug delivery technologies are essential for CRISPR-based therapy. They enable targeted delivery of gene editing tools into specific immune cells. CRISPR works at the gene level or molecular level, allowing precise molecular action. This improves safety and supports targeted CCR5 gene editing for HIV treatment. And lastly, foundation of CRISPR-based HIV therapy leads to CCR5 gene editing discussion. So basically, advanced drug delivery platforms enable CRISPR-based CCR5 gene editing as a potential HIV therapy. So in summary, modern drug delivery technologies provide the foundation of CRISPR-based HIV therapies. Because of these systems, CCR5 gene editing can be delivered safely and effectively, which will be discussed in the next section of this presentation. Thank you.
So, for the introduction of CRISPR-Cas9 for CCR5 gene editing, it is a revolutionary gene editing technology that can precisely cut and modify DNA sequences inside living cells. This allows scientists to target very specific genes responsible for certain diseases. In HIV therapy, which is still in the clinical experimental stage, CRISPR focuses on disabling the CCR5 gene. CCR5 is a receptor found on immune cells, and HIV uses it as a doorway to enter and infect the cell. By knocking out this gene, the immune cells become resistant to the virus. This type of approach is considered a targeted, genetic-based therapy. Unlike traditional antiretroviral drug that temporarily controls HIV, gene editing tries to modify the patient's DNA directly, offering a long-term or even permanent protection.
CRISPR is a powerful gene editing tool that works like a guided pair of scissors. It uses two main parts: a guide RNA, which searches for a specific DNA sequence, and the Cas9 enzyme, which cuts the DNA when the guide RNA finds the correct match. So in this case, the guide RNA is designed to match a section of the CCR5 gene. So the CCR5 gene is important because it makes the CCR5 receptor one of the main entry points that HIV uses to get into CD4 T cells. So the goal of CRISPR here is to disable CCR5 and block HIV from entering the cells. Next, when the guide RNA finds the CCR5 sequence in the DNA, it attaches to it, and this activates the Cas9 enzymes. So the Cas9 enzymes then cuts both strands of the CCR5 gene. This cut is shown in the diagram where the scissors appear inside the Cas9 protein. So next, after the cut, the cell tries to repair the broken DNA, but the repair process, called non-homologous end joining, is quick and often make mistakes. So these mistakes include insertion or the extra DNA letters added, deletion, DNA letters removed, or frame shifts, gene reading or order broken. So these mistakes are also called indels that disrupts the CCR5 gene. So the diagram shows this under deleting a gene and gene is disrupted. Okay, so because of these mutations, the CCR5 receptor can no longer form correctly. So the edited cells become CCR5 null, which means the receptor is absent from the cell surface. So people who naturally lack CCR5 are actually resistant to HIV. So CRISPR is copying this naturally protective mutation. So HIV needs two things to infect the cell. So it needs the CD4 receptor and the CCR5 receptor. So without CCR5 receptor, HIV cannot attach or fuse with the CD4 T cells. So these edited cells become resistant to HIV infection, similar to the natural CCR532 mutation seen in people who are naturally resistant in HIV. Another important part is that these edited cells survive better when HIV is present, so they multiply and start to replace unedited cells. Over time, the immune system becomes filled with CCR5 edited HIV resistant cells. This creates a long term protection and shows how CRISPR could be a used strategy for building lasting immunity against HIV.
Alright, so good day everyone. I am Alyssa, and today I will be discussing the advantages and limitations of the CCR5 targeted gene editing for HIV. So first, let's start with the advantages. It is a one-time therapy instead of the daily antiretroviral therapy. This approach aims to modify the patient's immune cells only once. Instead of relying on lifelong antiretroviral drugs to suppress HIV, gene editing tries to make the immune system naturally resistant to the virus. Once the cells are edited and reinfused, they can keep working indefinitely. Second, it is highly precise targeted editing. So, modern tools like the CRISPR can locate the exact part of the CCR5 gene and disable it. This precision reduces unwanted genetic changes and makes the therapy more controlled. The method mimics the naturally occurring CCR5 delta 32 mutation found in people who are naturally resistant to HIV. Third, it is a long-term protection against HIV entry. By knocking out CCR5, the virus loses one of its main entry doors. Edited cells can repopulate the body and continue resisting HIV for years. If enough edited cells survive and multiply, the patient can maintain a long-lasting resistance. And lastly, potential for a functional cure. So, a functional cure means the virus becomes undetectable and controlled without continuous medication. Gene editing could achieve this by creating a population of HIV-resistant immune cells, preventing the virus from reestablishing strong infection even after the antiretroviral therapy is stopped. Oh, and let us not forget the reduced dependence on donor-matched stem cell transplants. So earlier cures needed rare donors with a CCR5 delta 32 mutation plus a perfect match and carried risks like graft versus host disease. Gene editing avoids all that by using the patient's own cells, making the process safer and not dependent on finding a special donor. So the next we have the limitations. Number one, delivery efficiency remains low. So getting gene editing tools rather, into enough stem cells or T cells is still very difficult. Only a small percentage of collected cells successfully receive the edit. If too few cells are modified, the therapy won't work effectively once they are returned to the body. Number two, off-targeted editing needs monitoring. Even with precise tools, CRISPR can sometimes cut DNA in the wrong place. These off-targeted edits might cause harmful mutations. So, because of this risk, long-term monitoring and safety checks are required before large-scale use. So third, the cost is very, very high. So gene editing is expensive because it involves collecting patient cells, editing them in specialized labs, expanding them, reinfusing them, and monitoring them for months. This entire process makes the treatment inaccessible for many healthcare systems, especially in low-resource income countries. Next, regulatory and ethical constraints. So, permanent gene modification raises ethical issues, especially around safety, informed consent, and long-term effects. Governments require strict testing and clinical trial data before approving treatments. These regulations slow down deployment but ensure safety. Next, it is not necessarily effective against all HIV strains. So, some HIV strains do not rely on the CCR5 receptor. Instead, they use alternate co-receptors like CXCR4 to enter cells. This means that even if CCR5 is successfully edited and blocked, these variants can still infect the patient. Because HIV can switch the receptor it uses, CCR5 editing alone cannot guarantee full protection against all viral forms. Next, latent viral reservoirs and integrated provirus remain a hurdle. Even if new infections are prevented, HIV that already integrated into the DNA of the infected cells may remain. These latent reservoirs hide in certain tissues and stay inactive for long periods. Since gene editing mainly blocks viral entry and does not directly remove integrated provirus, these reservoirs can reactivate. This makes complete eradication of HIV still a major challenge. And lastly, it needs an advanced facilities. So this therapy requires high-tech laboratories, trained geneticists, and controlled environments. Many regions don't have the needed infrastructure, making widespread access very difficult. So it is currently limited to specialized research centers. Alright, and that is all for the limitations and advantages of the CCR5 targeted gene editing for HIV.
Okay, so now we're on to the article CCR5 Gene Editing and HIV Immunotherapy. So, current understanding, challenges, and future directions by Wang Liu and Xiong, published in Frontiers in Immunology, gives a detailed look at how CCR5 gene editing has become one of the most promising strategies for HIV treatment. The author explains that CCR5 is an essential co-receptor used by many HIV strains to enter the CD4 T cells, making it a major target for researchers who aim to block the virus at its entry point. A key example supporting this approach is the naturally occurring CCR5-32 mutations, which provide strong HIV resistance in individuals who carry it. So this real-world evidence inspired by scientists to try replicating the mutation through genetic engineering. The paper reviews different gene editing tools but highlights that the CRISPR or Cas9 as the most advanced and adaptable technology. So CRISPR allows precise disruption of the CCR5 gene, and study shows that edited immune cells can survive well, maintaining a normal function and demonstrate clear resistance to HIVs in laboratories and preclinical models. The author also discussed how researchers are now combining the CCR5 knockout with the various immunotherapy strategies, such as engineered immune cells or HIV targeting antibodies, to strengthen the antiviral responses and move closer to the functional cure. These findings show promising momentum in the field, with multiple studies supporting the scientific logic behind the CCR5 and demonstrating that gene-edited cells can genuinely resist HIV infections. The article emphasizes that this combined approach of gene editing plus immunotherapy may eventually reshape how HIV is treated, offering a more durable and personalized strategy compared to traditional antiviral drugs.
Despite the promising progress in HIV research, a complete cure for HIV still remains out of reach. One of the most exciting areas being explored today is CCR5 gene editing. However, there are still several important challenges that prevent it from being widely used. First, delivering gene editing tools safely and effectively into enough immune cells is still very difficult, even with advanced methods. Researchers struggle to reach a large enough number of cells for the treatment to work. There's also the risk of target mutations, where the editing tools might accidentally change other parts of the DNA. This can lead to potentially harmful effects. And another big concern is that HIV may adapt by switching to another co-receptor called CXCR4, which would make CCR5-based treatments less effective. So the review also emphasizes several broader issues, and we still need long-term safety data to fully understand the effects of these therapies over time. In addition, gene editing treatments are extremely expensive and access remains limited, especially in low-resource settings where HIV is most common. Overall, the article concludes that CCR5 gene editing is a powerful and scientifically supported direction for HIV therapy. But for it to succeed, researchers must continue improving delivery systems, ensure the long-term safety of edited cells, and combine gene editing with other therapeutic strategies. With these advancements, CCR5 gene editing may eventually become a key part of future HIV treatment.
So hello everyone, I'm Isa Marie Vasquez and I will be discussing to you guys the clinical relevance and ethics of CRISPR method. So in this slide, the clinical application of CRISPR CCR5 gene editing is a revolutionary approach with the potential to transform HIV treatment. So unlike lifelong antiretroviral therapy, this technology offers a one-time targeted therapy that can create HIV-resistant immune cells. So its application go beyond HIV with potential in other viral infections and future precision medicine. So CRISPR allows us to intervene at the gene level, opening new possibilities for clinical care. So next is the impact that this method has for the patients. So it could be life-changing because first, it is a simplified treatment. It is a one-time therapy that reduces the burden of daily medications. It also lowers the risk of long-term drug toxicity, and it also has durable protection. Potential for long-term resistance to HIV, improving the quality of life of patients. So this means that patients may enjoy better health outcomes and more freedom from the lifelong constraints of medications. Next is the relevance to the nursing practice. So as nurses, understanding CRISPR is essential because we need to build competence in gene therapy and precision medicine. We play a critical role in educating patients and guiding them to ethically true cutting-edge treatments. Third, nurses may also participate in future clinical trials and interdisciplinary care. So by understanding CRISPR, nurses can advocate for safe, effective, and equitable care. So despite its promises, CRISPR raises important ethical questions. So irreversible genetic changes require careful deliberation. Off-target effects could lead to unintended consequences, and equitable access is essential to prevent widening healthcare disparities. Lastly, societal debates around human DNA editing reminds us the broader implications. So CRISPR therapies are subject to strict regulations. It needs approval from organizations like FDA and EMA. It has a long-term monitoring to ensure patient safety. Its differences in international policy reflects cultural and ethical perspective on gene editing. So the future of CRISPR is quite promising. Its multi-gene targeting may provide broader protection. Its advanced delivery systems like exosomes or lipid nanoparticles may improve efficiency. Combining CRISPR HIV DNA excision with AI-guided precision could redefine viral disease management. So in conclusion, CRISPR CCR5 gene editing addresses the limitations of traditional art by offering a targeted, long-lasting, and potentially curative therapy. Nurses must stay informed to provide ethical guidance, patient education, and safe clinical care as this technology evolves.
Now that everything's been said and done, let's now move on to the questions. So 1. Can CRISPR delivery efficiently be improved? Number 2. Will the therapy be ever affordable? And 3. What role will nurses play in gene editing care? So again, we are the group 4, and here is the article or reference that we provided in this video right here. I hope that everyone learned something from today, and yes, thank you so much.